for coming today. Hopefully everybody's not too disappointed we didn't have snow. Monday snow was gorgeous. Everybody had a good weekend. Got to go to a Seder on Friday. Got to go to an Easter dinner on Sunday. Yay, hockey on Saturday. Next week will be our last event, and that's Dale Scholler from the Biotech Center and Nutritional Sciences. He's going to be talking about the possibility of getting more sleep and what that can do for your caloric intake and weight loss. It's all good news. All you have to do is get good sleep. Okay, relax. Yeah, man, ever since they took the good stuff out of NyQuil, man, I'm a hurt cowboy. Tonight at Wednesday night at the lab, Kevin Levin is going to be speaking on networks, the science of interconnected systems. I'm looking forward to that talk because I think he's going to not only use kinds of words that I understand, but hopefully show us some mathematical modeling for networks and systems and all that cool stuff. And then next week, Reba Lucan will be re-giving her talk that she gave here a couple of weeks ago. So if you want to send any friends to that talk, she'll be at Wednesday Night Lab. And then in 2 weeks at Wednesday Night Lab, Dan Laufer with Wisconsin Fast Plants will be talking about the collaboration between Ukraine and UW-Madison and the Fast Plants program. And he sent me an email a few weeks ago after the Russians invaded Ukraine and asked if he could give this talk. So I'm very happy to accommodate that. And then next week, of course, and if you want to start this week with ideas for the fall, that would be great. And we'll collect ideas for topics for the fall also. So we'll be done on next week on the 27th. All right. Yay. This week, we have Vanessa Leone with us. I'm going to ask you the 5 questions.
Okay.
Where were you born?
I was born right here in Wisconsin in Menominee Falls.
Whoa. And where'd you go to high school?
Hartford Union High School. I talked about that earlier.
Very good. And where'd you go to undergrad and what'd you study?
I went to UW-Madison. I studied animal sciences. That was a department at that point.
Yep. Now the department is animal and dairy science. Yep. Yep. And then where'd you go for your advanced degrees? What'd you study?
So I did my PhD at UW-Madison as well. I studied with Mark Cook, who maybe had spoken through this series before he passed away in 2017. And then I was at University of Chicago for a postdoc and instructorship before I came back here as faculty.
And were you in the Department of Animal Science or the Department of Dairy Science at the University of Chicago?
I was in neither. I actually took a little jaunt into the realm of medicine and clinical medicine, trying to learn more about how my science can be applicable to human health and disease, and where I learned all about the microbiome.
Very good. And when did you start your faculty position here?
Yeah, so I'm in my 2nd year here. So I started right in the middle of the pandemic. So it's been really slow going to get up and running. So I think I was supposed to start July 1st of 2020. I didn't end up arriving until October of 2020. So I'm in my 2nd year now here.
And where's your lab?
So I will describe a little bit about that. I actually have labs in 2 spaces. So I actually have the old Cook lab in the Animal Sciences building on campus on the 10th floor. So you can oftentimes find me there. But then
view. Are you facing north to the lake or?
Well, I don't know if this group is familiar with Ralph Albrecht, but I actually hijacked, since he went emeritus, I hijacked his office, which has a beautiful view of the lake. But then I spend a lot of my time over in the new building, which I'll show you on the next slide, which is the Meat Science and Animal Biologics Discovery Building, which is the newest, I think right now it does stand as the newest building on campus. And, you know, is a huge investment between the state and industry partners and their taxpayer partners to really bring and elevate meat science and have a new effort called Animal Biologics that I'll describe briefly before I talk to you about my main research focus.
Great. Well, I appreciate you speaking today, and would you please join me in welcoming Vanessa Leone, who brings it to PLATO.
Well, it's a really big honor and pleasure to be here. You know, starting a lab in the middle of a pandemic, moving in the middle of a pandemic is challenging. While I had spent about 10 years of my life here in Madison, I had a 10-year hiatus as well. So while a lot of things stay the same, so much changes. So it's really nice to be out and about in the community to meet everyone. Today, I'm going to talk to you about some of the stuff and work that I started at University of Chicago during my postdoc and instructorship, and that I've transitioned here to UW with me. So I will be showing you some primary data. You know, I'm just hoping that you can walk away with some pieces of it or ask questions about it. I would really like for this to be more conversational than anything. And, you know, with that, I think we'll just kind of get started. If I can get my computer to agree with me. So as mentioned, I am part of the new effort on campus called the Meat Science and Animal Biologics Discovery Program. So this is the new building on campus. There's actually a meat market, amongst other things, in the building. So it's called Bucky's Varsity Meats. And we actually, for animals that are processed directly in the facility, that those products make it right to the store. So a lot of what we have there is fresh meats. We also have just an amazing butcher in our department, and he's been really trying to ramp up on all the products that he can offer. So I would encourage you, if you're ever bored, to come and stop by and check it out. Oh, great question. So it's on Observatory Drive. So those of you who are familiar with the natatorium, it's right across from what used to be the Nat, and now is the brand new gym that they're building too. So yeah, it's right inside the door there. And so this building actually has a full USDA inspected processing facility in it. So it really allows us to do some unique things under proper inspection as well, to work towards goals of, improving food safety, processing, product development. It's a really cool place to be. And so my lab is small right now. We've started very slow. I have a postdoc in the lab, Dr. Samar Tolba, who is here from Egypt. She did a joint PhD between Cornell and Zagazig University in Egypt. And so she's here. She's our resident poultry and nutrition expert. Jake Hermanson was actually the first member of my lab. I built my lab a little backwards, some people would say, and he's actually a graduate student. So he's a second-year grad student, and he's a nutritional sciences graduate student. And then we just recently added a researcher or technician to our lab, Lori Sand, who has a master's degree actually in arts. And so she has a journalism background, which has been phenomenal for someone who's writing grants like I am, trying to get more funding. So, all together, we're really trying to build a team. And we have a slew of undergrads, so many that I didn't feel it was right to just selectively choose a few to put on the slide. So what is my research focus? Well, when I came from University of Chicago, when I should say and back up and say, when I was here as a graduate student, I was focused on poultry nutrition. So working with Mark Cook, he was a poultry immunologist, or chickenologist is what he self-prescribed himself as. And we were really interested in looking at how dietary supplementation could impact avian embryogenesis and development of chicks. And so I took a lot of nutritionally focused courses during my undergrad and graduate work. And right about the time I was looking for my next step to postdoc, the microbiome really started to reemerge as being this important component of health versus disease. And we knew and we've known for years, really, and centuries, that diet and our environment can impact microbes, either in a beneficial way or a negative way, which we'll talk about today. And I thought maybe my nutrition and poultry background could be interesting to apply to the microbiome space. So I went to University of Chicago to learn all about microbiome. And so a lot of my work focuses on understanding how gut microbes impact metabolic health in the context of lean versus obesity, and identifying qualities that can kind of describe what is a lean microbiome relative to what is an obese microbiome. And then the other hat that I wear is part of the animal biologics effort. So on campus, we have this big focus where we want to actually make meat the byproduct and all other components useful for human health. Or animal health, right? So there's a lot of product or portions of the animal, probably between 40 and 60%, that are actually discarded at the time of slaughter. And so we think that because all of these components that are in this animal are meant to support life, right? So we are wondering, can we kind of isolate some of these components and use them as human medicine? One really great example that is a biologic, what we call a biologic, is heparin. So I'm sure many of you have heard of heparin. Do any of you know what the predominant source of heparin is? Yeah, it's porcine mucosa, exactly. So many people don't understand that. There is no synthetic analog of heparin available. So about 90% of our heparin supply comes from porcine mucosa. So it's the lining of the intestine. Yeah. So think about all of the other components that might get discarded, and can we isolate some of these novel molecules to sustain or promote health? So with that in mind, I really want to give you an overview. So I'm going to kind of describe this role of diet and microbes and their role in metabolic health. I'll present to you some of our initial findings that helped us to connect these concepts of how microbes impact circadian rhythms. And I'll tell you a little bit about what circadian rhythms are and how did we really get here, and then go into some of my advanced findings, which, as you mentioned earlier, just came out online. They were just published on Monday, I think. So we're really excited to kind of delve into some more mechanistic roles here to see how, not only can we break this system, but how can we maybe fix it? And then hopefully we can discuss about some key takeaways. And I'm interested in learning from you also how we can apply this moving forward. So I don't think this is any shock to anyone in the audience, but the prevalence of what we call new age disorders has increased over the past half century, and even maybe a little bit longer at this point. So when we think about even self-reported diseases like obesity and even type 2 diabetes, starting back in the 80s, there was relatively low incidence here for disease. And this has just dramatically ramped up, where, when we look across the United States, that some of these states are actually reporting, 26% of the population is obese. And diabetes, type 2 diabetes especially, is marching right along with obesity because, these diseases are multifactorial and complex. But I always say the situation is getting worse when they change the colors on these maps. And so you can see even from 2018, now there's certain states that are recording over 35% of the population as being obese. And last night I looked up the most recent map from the CDC, because these are always coming out and changing. And now we actually have some states that are getting close to reporting greater than 40% obesity in their population. That is a major problem because we don't really understand how to fix this, right? Aside from major lifestyle interventions, there really is not a magic silver bullet to resolve obesity. And why is this? The disease is multifactorial and complex. There's no doubt about this. There's definitely a genetic component that comes into play, but not everybody who has a genetic risk factor will develop obesity. So how do we tease that apart? We know that there's a number of environmental influences that can impact our genetics to promote the development of these complex metabolic diseases. And some that I've been most interested in, as you can imagine, based on the title of the talk, is this combination of how nutrition and diet can impact gut microbes. And with this, what I actually added to this slide was sleep and sleep disruption. Now, I don't focus mainly on sleep. I focus on circadian rhythms. Some people will argue sleep and circadian rhythms are not identical. They are not interchangeable. And I do agree with that. But I think to really grasp onto what circadian rhythms are, sleep is such a huge factor that I would be remiss if I didn't have it on this slide. Yeah.
I noticed you have social status there, but what you don't have that I think is, in my mind, is important is, and maybe you have it in social status, is economics.
Yeah.
Because if you're poor, you only have access to the worst food.
That is so true. We talk about, food deserts versus, food swamps. I think that's a newer term that,
Well, and even if you have access to the store, I mean, it's cheaper to buy your kids soda than it is milk.
Absolutely. Yeah. So economic status, social status, I think In this specific figure, they're actually talking about them being interchangeable, but I totally agree that there's some differences there that could influence this.
Is there a way that you can drop that bar across the top?
Oh. Yeah, I can see if I can move it. Let me see. I'm going to put it over the title because I think we all know why we're here. But no, you're exactly right. And I think the next slide kind of gets after this a little bit more. So when we think about our population and who we are as humans, we really were a bit of a nomadic population, right? We had a lot of hunter-gatherers, and, you know, even until 100 years ago, highly processed foods were not as readily available. So we kind of had this big shift of, you know, having less access to less calorie dense food, to going into this situation where we have high access to highly processed, high calorically dense food. And I think this is exactly what you're talking about, where it's a much easier to grab, you know, a Big Mac at a cheaper price than it is to sometimes go and find some very nutritionally valuable and rich food sources. So what does this mean as far as the microbiome? Well, even though this study now is from 2016, it is still, or I'm sorry, 2010, it's still one of my favorite examples of just kind of a very clear, visually pleasing way to think about how diet especially can impact microbes. So this was a study that was actually done by a group over in Italy, where they were looking at children who were raised either in Burkina Faso, Africa, or the European Union, specifically Italy. And before they even looked at additional factors that were affecting the microbiota, they profiled the microbiota. And so how do we profile the microbiota? Well, we can actually use conserved genes in microbes themselves that are conserved across all bacteria, but have small enough differences that we can pick out whether or not, let's say, they're a lactobacillus or they're a bifidobacteria species. So this is how they were using this next-gen sequencing technology, examining 16S specifically to profile these microbial populations. And just by looking at these pie charts, which describe the relative abundance of these microbes in the community, you can see just based off of the colors that they're very different, right? And so when they actually went to look to see what are factors that are driving these differences, one of the major ones that came out was the dietary intake. So not only were there strong differences in caloric intake between these two groups of children, but also the distribution of calories across the major nutrients, protein, fat, and carbs, was actually quite unique as well. And so you can see that the children in Burkina or in the EU are eating quite a bit more fat, and they're also taking in quite a bit more carbs. And some may kind of question that and what that means. Well, I would argue that the children in Burkina Faso, Africa, are probably taking in quite a bit more fiber, whereas those in the EU are taking in quite a bit more simple sugars. So like when you're talking about soda, for instance, the children in the EU are definitely more exposed to things containing, simple sugars. So this dramatically associates with how the microbes themselves are shaped in our intestines. But association doesn't mean causation, right? So we have to think about it as researchers, how do we delve in and really identify causation and what's driving this? Well, back in the late 90s, early 2000s, Jeff Gordon down at Washington University kind of reemerged a technology that had existed for quite some time called Metabolics, or germ-free. And he was using this to kind of probe these host microbe interactions. And what he found was that mice, for example, who are raised in a standard environment, who always have exposure to microbes that are exposed to a high-fat diet, will develop obesity. But now their germ-free counterparts that are raised in the complete absence of microbes, and they're literally like, I don't know if y'all are familiar with Bubble Boy, but these are essentially what these mice are. They're raised in bubbles and they're exposed to all sterilized materials. They can live full, happy lives, but they're never exposed to microbes. Now they have very unique physiology without the presence of microbes, but it allows us to kind of ask questions. Well, what happens when you don't have any microbes present? And so when we put these mice onto a high-fat diet, they actually remain lean for the most part. So they don't develop obesity. So this tells us that for at least in regard to diet-induced obesity, we need to have microbes present in order to drive that to occur. So since these initial findings came out, they've been recapitulated many times, and there's been many mechanisms that have been put forth for why this occurs. And so, there's this idea that the types of microbes you have in your gut that are driven by the type of diet you take in can actually alter energy extraction from the diet. So maybe there's just microbes in your gut that can get more out of those simple sugars, for example, which changes our energy balance. It could be that these microbes impact gut permeability. So, we always talk about low-grade inflammation. Well, perhaps these changes in microbes can actually contribute to low-grade inflammation, both locally in our intestines as well as in the periphery, like in our livers or in adipose tissue. There's some talk about how even microbes can impact gut-brain axis and our behavioral outputs. And so that's actually some work that I'm doing separately that I won't be talking about today. But then we really ask the question, are there novel mechanisms for the role of how gut microbes are communicating with us? And so I had a really fun advisor for my postdoc down at UChicago, and he challenged us. He said, okay, look, we've got all of these sequencing technologies available to us. Why don't we just look at gene expression between our germ-free mouse that doesn't have microbes as compared to our conventionally raised mice, or what I call specific pathogen-free mice that have a full milieu and complex community of microbes. And so this is what we did. And so this may seem a little bit meaningless to you, but it's a way to look at relative expression of certain genes between one group of animals versus another. And so what we saw was that across all of these tissues that we looked at between germ-free and our conventionally raised counterparts, that genes involved in circadian rhythm signaling kept popping up. And we thought, we were not circadian rhythm people at the time. So what is circadian rhythms? Well, digging into that, when we kind of dissect that word circadian, it literally means circa dias, or around a day. So what is happening over a 24-hour period? And we know from work done by other people that the circadian gene network itself, which is a complex network of, feed forward and feedback loop of interactions between specific genes to drive oscillations or day versus night rhythms in specific processes that are occurring in the body. And so this is a very complex network that you certainly don't need to know, but this is really what we consider to be the metronome of life. So this helps us to understand when it's light versus dark, when we should be sleeping versus when we should be eating, and even at the molecular level, when we should be extracting energy versus storing energy, as well as even repairing DNA damage. So disruptions to our circadian systems can also lead to things like cancer, for example. So we wanted to know, well, gut microbes haven't really been considered as part of this network previously. So how are our gut microbes, contributing to this? And we know that if we genetically manipulate, not humans, of course, but mice, for example, that we can disrupt their circadian rhythms and impact health. We know that people who undergo shift work or experience sleep apnea or jet lag, this disrupts their circadian rhythms and can lead to disruptions in metabolic health. And simply by taking in a high-fat diet, at least in rodent models, that alone is enough to shift our circadian rhythms. So how do microbes fit in? Well, in order to dissect that, we really felt that we needed to kind of use all the tools available to us, right? So we needed to use some in vitro cell culture systems where we could really manipulate very carefully certain aspects of this system. And we could also use rodent models. So I told you about the germ-free mouse. So how could we use that mouse to really identify the importance of this host microbe crosstalk? And ultimately, our goal is to understand how microbes influence not just animal health, but also human health as well. So our first paper that we actually published in this regard kind of went like this. So we really felt and feel that gut microbes are there to sense dietary cues that can be translated by our body into outputs that help to maintain circadian networks. So when all systems are running properly, your brain clock, your liver clock, your intestinal clock, your adipose tissue clock should all be coordinated and communicating appropriately. But when this becomes disrupted, all of the clocks can be running on their own time. And you can imagine that that's probably not good, right? Like it's a little bit like looking at a highway, right? When all of a sudden you've got rush hour traffic and everything's all happening at once, it can kind of result in what we might consider to be futile cycles, right? So we think that the gut microbes are actually in our bodies to help us sense when, how much, and what we're eating. So in animals, this is all in animals, if they were eating a low-fat, high-fiber diet, we saw that the microbes, both in who was there as well as the relative abundance, exhibited a day versus night pattern, or what we call an oscillation. And this oscillation was both in who's there as well as the metabolites or molecules that these microbes are making that then we absorb into our own bodies and can influence the clocks that are located both in the brain and in our peripheral tissues like the liver. So you can see that all of these signals are very nicely coordinated and ultimately results in wellness. But when this becomes disrupted by high-fat diet, for example, the situation changes. So this is when now maybe we're taking in a high-fat, high simple sugar diet. We see that microbial oscillations are actually dampened in this case. And this could be due to a number of different things like time of feeding, for example. So if you feed a mouse a high-fat diet, usually they consolidate all of their feeding to the dark cycle. But if you give them high-fat, they say it's party all the time, and they will eat during the day as well as during the dark. So that actually ends up in a dampening of the rhythms in the gut. So that changes the gut clock. The metabolites that these microbes are making changes and disrupts that specific signal. And now our host tissues are seeing these metabolites at very different times that maybe even when they shouldn't be seeing them. And this can result in disruptions in our own clocks. And this can contribute to the development of obesity. But now this can also go the other way. So for example, if we experience jet lag here, or we genetically modulate the clock, again, in animals, or put them through an experimental jet lag protocol, that we can also obviously then disrupt our own tissues, but this can feed back onto the microbial signals and can disrupt the rhythms that way too. So this is definitely a two-way street that we're talking about. And ultimately, this can also contribute to metabolic disruption. But it's not all lost. There are ways to fix this. So one of my colleagues at UCSD, Amir Zarenpar, did timed feeding where he fed a high-fat diet to mice only during the dark cycle when they should be eating, because mice are nocturnal, whereas we are diurnal. And when he did that, what he saw is that even though these mice were eating high-fat diet, they had a consolidated feeding period only to their active phase, and he could restore some of the microbial oscillations, not all, but some. And ultimately, this was able to restore host circadian functionality and prevent the development of obesity. So it's called time-restricted feeding, or some of you may be familiar with the term intermittent fasting, right? So this is essentially what Amir was doing with his mice and showing proof of concept. And now his mentor, Sachin Panda, is the one who has put forth some of these apps that we can put on our phones to perform intermittent fasting and have timers and all of these other things. So it's a really interesting phenomenon. But this is great that we can do it in mice, but what does it mean for humans? And how do these rhythms and microbes and the host really show up in humans? Well, there's been some simple studies to show that this does actually happen in humans. And what's the biggest challenge of doing human work when we're talking about microbiome? We're really reliant on testing stool, for example, and not everybody can produce a stool sample every 4 hours. So how else can we do this? We can actually do saliva, and we can look to see what does the microbiome look like in your mouth, and we can routinely swab that, for example, and we can look at rhythms in our saliva, for example, as far as the microbes are concerned. So very non-invasive way to look at microbes. So in this study, this was actually done by a group over in Spain in collaboration with a group at Harvard. They actually took 10 healthy women, and they had some of them eat early, and then they had some of them eat late, and then they did a crossover. So they switched them over. And these, again, were healthy individuals, so I can't really say too much about what it means during obesity, for example. But what they did find was that they could sample the salivary microbiota at least at 4 time points over a 24-hour period. And what they saw was that there were really unique day versus night patterns or diurnal patterns in microbes that were observed based on the time of eating. So whether or not you had early eating versus late eating. And so there was an inverted pattern depending upon the time of day that feeding was occurring. And eating late, so, those of us who maybe have a midnight snack, if you will, had increased members of the community, microbial community, that were associated with inflammation. So it seems that earlier eating can actually be beneficial for our microbes as well, from a human perspective. And again, this is associative. Now, this study, I wish I could have taken part in because I'm a little bit of a chocoholic. But this same group, actually, just in 2021, they were looking at chocolate intake, because chocolate gets a bad rap, right, as being maybe not so beneficial for our health. But what they did was they had, again, a group of, I think this was premenopausal women that were taking in either no chocolate, or they had morning chocolate or evening chocolate. And we are not just talking about a piece of chocolate, we are talking about a chocolate bar. So this is 100 grams of milk chocolate over 28 days. Again, I want to be in this study. But what I think is really interesting is that depending on whether or not the people were taking in, no chocolate, morning chocolate, or evening chocolate, there were some changes in microbial byproducts, we'll call them, and the patterns of those. So here we're looking at something called short chain fatty acids that microbes make. There's things that, like, we call them butyrate or acetate, propionate. These molecules have been shown to be beneficial for host health. Now, if you're a ruminant person, you know all about butyrate and what maybe we call volatile fatty acids. So if you're, into dairy cows or beef cows or sheep, they make a lot of volatile fatty acids that are beneficial for health. So this is kind of a marker for what the microbes are doing. And so, you can see that there's not so many changes when we consider acetate, but now when we look at propionate, no chocolate is actually causing less.
Can you tell what the colors are?
Oh, yeah. Absolutely.
I can't see what the words are saying.
Yeah, sure. So no chocolate is green, morning chocolate is blue, and orange is oh wait, no, I have this backwards. Evening chocolate is blue, so blue nighttime, right? Morning chocolate, so sun, is red, is orange. Sorry, I'm acting like I'm colorblind, but I'm not. So you can see that no chocolate here in the green is causing less amounts of propionate being produced relative to either evening chocolate or morning chocolate. Well, that's a good question. So this is just looking at profiling of microbes. So basically what we can see is that no chocolate, this is what we call a principal coordinate plot. It's a little bit like, I don't know, I would say this is like Sesame Street, like trying to figure out what looks like something else, right? So spatially, how closely related they are means that they're more similar. So no chocolate clearly is very similar to one another's, but the microbes from either morning chocolate or evening chocolate were segregating depending upon the time of chocolate intake. So I think early chocolate, probably better, right? And so this is just, again, kind of looking at that further, and then looking at, for these short chain fatty acids that are produced, acetate in particular, these are the relative abundances of certain microbes that are associated with making these short chain fatty acids. So like ruminococcus, for example, is more elevated regardless of whether or not you have morning or evening chocolate relative to no chocolate. But the story becomes a little bit different when we consider something like Lactobacillus, a different type of microbe, where now maybe we see a little bit more spread depending upon time of day of eating chocolate relative to no chocolate.
What does that mean?
So what this is telling us is that taking in chocolate, whether it's early or late in the day, is shifting our microbes, right? So it's just saying, like, if you don't have any chocolate, your microbes are going to look like this, and this is what the community membership is going to look like. If you do have chocolate, you're going to see a shift and a change in those microbes.
And what does that mean?
Well, that's a good question, because actually in this study, especially the women who were eating morning chocolate actually lost weight.
Oh.
Yep. I know. So it's kind of interesting.
What is the unlactose race?
Yeah, so that I know this is just kind of a way for how we call these microbes. So the microbial names are constantly changing. So unlactose is probably a specific group of lactose relative to others. So it's just a little part of the name.
Perhaps a monkey wrench here. The woman who eats the chocolate in the morning are eating for the rest of the day, whereas the evening is not. If I had morning chocolate, I probably wouldn't be that hungry for lunch or snacking.
Yep.
So does this skew and this make the results somewhat misleading?
Yeah, no, I agree with that because I think they did food intake recordings of these people using something called meal logger. And they did find that the women who had morning chocolate ate less throughout the day, right? So you're right. That's the time of this high calorically dense food can change your eating habits throughout the day. So
If they eat at night at all,
the individuals or,
the early morning chocolate.
oh yeah, they still ate throughout the day. Yeah, they weren't forcing anybody to eat consolidated meals.
So they,
Yeah.
they might have been eating midnight.
It's possible. It's possible. Yeah. Yeah, I don't have that data specifically. But yeah, I think it is really interesting though that timing of chocolate intake, can ultimately impact maybe some of the outputs as well.
Any other high fat food substitute for the chocolate?
It's possible. Yeah. I mean, what are you thinking? Something like cheese? Because that's another one I wouldn't cry about.
It doesn't have to be chocolate.
Yeah, I don't think it necessarily has to be chocolate. It just happens to be what they were studying, but they didn't compare it to anything else. It was just no chocolate or morning or evening chocolate. So yeah, it's a good point. I will say that there is interest in trying to understand how maybe what we would consider other high fat products like cheese, for example, can impact the microbiota as well. Because cheese, and I'm certain I don't want to give milk a bad rap because it's got amazing qualities to it. But cheese has, it's a really rich and complex food. And so, it is interesting to try to understand how fat from different sources or like avocados, for example, right? Very rich in fat, but they're quote unquote beneficial fats, right? So it'd be interesting to kind of look at it in this context too. Yeah, it's a good question. I don't have the answers, unfortunately. So here we don't have any overt changes in weight, but we do know that morning chocolate can enhance lipid oxidation. And then evening chocolate can alter, I guess they were looking at sleep episodes too. So they actually had these people in an in-lab session too. So from my perspective, after kind of seeing this, I really wondered, is feeding by mouth required to drive oscillations in microbes, right? So is it really only time of day dependent? And so with a group here at UW-Madison, Kenneth Kutz's group, I think he is also emeritus at this point, but he was a surgeon here. He was actually one of the only ones in the world doing parenteral feeding in mice. So we didn't do our studies in humans, but we did them in mice. And so parenteral feeding means constant infusion of nutrients into the periphery as compared to enteral feeding, which is intake by mouth. And so we were interested, if we did this in mice, what would happen to the microbes and the rhythms. So what we did was we did a constant infusion of parenteral nutrition into mice as compared to enteral intake of just a regular low fat, high fiber chow diet in our rodent counterparts here. And we were actually pretty shocked because what we found was that parenteral feeding, so this graph here is just showing the relative abundancies of specific microbial community members based off of the color of the bar over a 24-hour period. And so you can kind of see here based off of the colors that there's still rhythms that are evident over a 24-hour period. So even though the microbial community is unique relative to enteral feeding or intake by mouth, there's still oscillations evident. So this tells us that there's other host cues that might be able to contribute to driving rhythms in certain members of the community of the microbiota. And this is important for us to know because, time of feeding is one thing, but we kind of want to figure out not everybody can sustain a specific timed feeding regimen, right? It does not fit into our lifestyles. So we want to kind of find other ways that maybe we can kind of have our cake and eat it too. So how can we manipulate the system? And so that's really what I've been trying to understand is what are these host-derived factors that exhibit a day versus night pattern, and how do they communicate and provide this feedback with the microbes themselves? So we've been looking at what we call antimicrobial peptides, which is something our body makes to try to keep pathogens at bay. So for example, like Listeria or Salmonella, for example, our bodies can actually make certain antimicrobials that can eliminate those bad guys from the community. And surprisingly enough, we saw that under parenteral feeding conditions, which is this red line here, that over a 24-hour period, there was no more rhythm that was present. It was only observed in our enterally fed animals here indicated in the blue line. And at the same time, another group had shown that if they depleted microbes with antibiotics, for example, so here in the yellow, they also lost a rhythm in this specific host-produced antimicrobial peptide. So this really got us thinking like, wow, it doesn't seem to be really related to anything the host is doing per se, but if we change the microbial community, we can change this factor being made by the host. So we really started to ask the question, like, is it our clocks that are driving this, or is it the microbes that are driving this? And how do we tease that apart? And so it kind of led to our major question in the paper that we just published is, what are these dominant versus secondary drivers for these microbial rhythms in the intestine? And so we've really been trying to dissect that out. So is it only, our brains seeing these light versus dark cues, for example, in the environment, or is it the time of feeding, or is there a direct communication between our clocks and the microbes that are driving these rhythms? Or really, here's the more complex one, is it a little loop, which I'll say it's probably a loop.
You're talking about stomach rather than the intestine?
Just throughout the intestine. Actually, one region we focused on in this particular study was the ileum. So it's the most distal part of our small intestine before the colon. And so we went back to our germ-free versus conventionally raised animals here, and we fed them either our regular chow low-fat diet or our high-fat diet. And we looked at the patterns of expression of our clock genes, which are indicated here by BMAL1 and PER2. So these kind of govern that day versus night rhythm. And these all looked pretty much the same across all the groups. So not much difference, which was confusing to us because we thought maybe this antimicrobial peptide we were interested in was governed by the circadian clock, but it actually is not. So if we look at our conventionally raised animals fed a low-fat diet, you can see this beautiful rhythm here that's occurring over a day versus night light-dark cycle. But when we feed these same animals a high-fat diet, we lose that rhythm again. And our germ-free mice never had a rhythm to begin with. So it really told us that these diet-induced microbes are in charge of this situation. So that's really interesting because it kind of uncouples all of these pieces. And so now we've just been trying to figure out, well, who then is in charge of this situation? And so we've kind of validated these things using pretty pictures. We can actually use histology to show that, only our regular chow-fed animals have elevations of this antimicrobial peptide indicated by the yellow, which probably isn't coming through very well in here with the lights. But there's only yellow here in the middle of that cycle, like I showed here, in the intestine itself. So we know this is not just happening at the gene level, it's also happening at the protein level too. And so we've gone on to show that using a number of different approaches. So validate, right? And now we've also been interested in, okay, if that's true, what's happening with the gut microbes? So you asked about the region of the gut that we were interested in. And so this kind of indicates that here in the distal ileal portion of the small intestine. So that's where all of our samples were collected from. And so again, looking at our principal coordinate plot, you can see that high-fat microbes in the distal ileum look very different from our regular chow-fed microbes. And this is occurring at multiple levels depending on how we look at it. So you can see that the red is always different from the blue. And then we really wanted to break it down to say, okay, well, our communities are changing, but what's happening with these oscillations or these, rhythms in the intestine? And so just like we showed in our previous study, even in the ileum, we were losing oscillations or dampening these oscillations. So, about only 5% of the communities oscillating under high fat feeding, whereas like 15 to 20% is oscillating under regular chow feeding conditions. And we wanted to know, are specific members kind of driving this phenomenon? And so we went back and did correlation analysis. So again, this is only association, but we needed to kind of find some leads to follow. And so when we looked at our Reg3 expression, we could see that there was positive correlations with certain community members, namely lactobacillus. So that's a lot of what we know is a probiotic type of microbe in the gut. But on the flip side, when we had high fat feeding, we noticed that there were other microbes that were negatively correlating. So they were very enriched in high fat feeding where Reg3 or this antimicrobial peptide expression was very low. So we wanted to know, well, okay, again, this is just association, but can we really go after this and figure out, are these drivers or not? And so with this, we kind of had these interim conclusions. We knew that this antimicrobial peptide was independent of the clock, exhibiting diurnal expression. We knew that feeding high-fat diet could eliminate these oscillations and change these correlations between this antimicrobial peptide and its diurnal behavior. We knew that high-fat was shifting the microbial oscillators, and that these, diet-dependent taxa were correlated with diurnal Reg3 or this antimicrobial peptide expression. But that's just association. So we really wanted to use some tools to move beyond association and really look at causation. So, how can we tease apart whether or not these different microbes promoted by diet are actually engaging with our host cells to drive antimicrobial production? And so, we were able to kind of prepare what we call like a cell-free lysate. So we just take intestinal contents and eliminate all the microbes and particles that are coming from the diet and make kind of like a poop soup, if you will. And then we can also isolate the same condition media from microbes that we grow in culture ourselves. So we grew Lactobacillus rhamnosus GG, which is in a lot of your yogurts. And then we had a couple high-fat microbes as well. So Peptostreptocacaceae, which is a mouthful, I'll call it Pepto, and a Clostridiaceae species as well. And we prepared what we call conditioned cell-free media. Now, we used some really novel techniques and approaches. So what we did was we made mini guts. So intestinal cells are classically difficult to grow in culture. So what we did was we popped out what we call stem cells from the crypts of the intestinal villi, and we grew those out. And what happens is they make what we call mini guts. And so mini guts do pretty much, they have the same cell distribution as our intestine. They're pretty much functionally similar to our intestine, but they're about yay big. So we can actually use these to probe, what does the small molecules that are made by these microbes actually do to the intestinal cells themselves in culture. So kind of a medium throughput assay, if you will. So by doing this, we took our ileal poop soup and from our regular chow fed and our high fat fed animals, and we exposed these mini guts to that. And we saw, sure enough, that our Reg3 or antimicrobial expression was really only observed when we had our regular chow, what we call lysate, and not so much with our high fat. And this was statistically significant. And we saw something very similar with our specific individual microbes of interest. So really only Lactobacillus rhamnosus, which was correlating in vivo in the animals themselves, were able to induce this Reg3. And this wasn't happening with our high fat fed microbes. So this was really saying that the microbes that are selected by diet are driving these outcomes, right? And so we went on to show that it is small molecules. So anything less than 3 kilodaltons coming from Lactobacillus, for example, is able to induce this. So that could eventually lead us to target molecules that we can isolate and use as maybe a potential therapy down the road, very much down the road, but it gives us some clues, right? And we knew that this was also important because if we took even these very small molecules and we heat treated them, which can inactivate them and disrupt the proteins, that we lost this activation pattern that we were seeing. So it means that these small molecules are less than 3 kilodaltons and they're very sensitive to heat. And so we can heat and activate them to prevent their capacity to induce this beneficial antimicrobial peptide. So this was really exciting for us. And there's more information in the paper that just came out recently too. So, this says that diet-induced microbial communities can differentially alter this antimicrobial expression in vitro, and individual bacteria seem to be involved in this cross communication. And it really seems to be specific small molecules that are made by diet-induced microbes that can induce or suppress this activity. But this is great to show it in these mini guts, but now we need to go back to a live animal, right? So this is this back and forth communication on the bench, back to the animal, back to the bench to try to validate and verify what we're seeing. So again, we went back to our germ-free mouse. We were able to feed it the different diets that I have shown you throughout the talk, and we could actually add a single bacteria. So we actually added lactobacillus all by itself to see what was it going to do as far as this antimicrobial protein expression. And so that's exactly what we did. So we took these animals off at 2 different times a day where we expected this antimicrobial peptide to be most highly expressed at ZT10, which is right before the lights go off in the facility. And we saw the highest expression here, and it did exhibit diet dependency. So this says that, the type of microbe that you associate an animal with and that's present in that community interacts with that diet and then can impact our own gene expression related. Oh, Zeitgeber, that's a good question. Yeah, I haven't really called it that throughout the talk. So Zeitgeber is kind of what we use to indicate an entrainment signal. So for example, light is a Zeitgeber, meal timing can be a Zeitgeber, changes in temperature can be a Zeitgeber. So they're really entrainment signals for our circadian system. Yeah, so all of these animals are held under a 12-12 light dark cycle. So the main entrainment cue here would be light. So kind of taken together, it seems that the type of diet is our major primary driver, right, for what is shaping our microbial community membership. But this dietary intake is really important because it's driving oscillations in key members of the community that can engage with us to drive expression of very important molecules in the body to keep that microbial milieu in check, really. And then this kind of, can feed back onto other members of the community to keep that community, working together in a beneficial way for us, really. And this can result in a lean animal, but we think that when you take in a high-fat diet, this very much changes the situation because you shift the microbial community membership. You lose certain community members that can then not engage with us, and that can actually allow for other microbial community members to expand and maybe do something that's not as beneficial for our bodies and can ultimately result in obesity. So that's kind of where we're at with this right now. We've done a couple other things, but really what we want to drill down on is trying to understand what these small molecules are that are being made by these lactobacillus that are engaging in this. So we could harness that maybe to promote health. But, what I think is really interesting, and hopefully what you walk away from is like timing is everything, right? And time stamping and understanding your chronotype, for example, is really important. And we have all of these large cohort data sets now where it's just, thousands of patients or subjects have contributed their stool, in order to try to understand how and to define a healthy microbiome. But one of our colleagues in this area did something really neat. So there was a very large cohort of people that had healthy and people with type 2 diabetes and obesity, all mixed in. So it was like thousands of people. And so they did this same thing that we've been doing. And, what they were able to do is sort of take these microbial communities that they were detecting in stool as compared to all of the host data that we had, or health data from these people. And what they ultimately saw was that they could bin them into healthy versus people with type 2 diabetes. And they noticed that the rhythms in the microbes that they could kind of reassemble because they had time-stamped stool samples. So they knew when everybody had deposited their sample, they could reassemble rhythms in the healthy people. And those rhythms were absent in the type 2 diabetes people. And so then they were able to use, some very novel approaches using machine learning to sort of identify this arrhythmic microbial signature that then they could apply back to their population, and they could actually bin people based off of this signature into healthy, maybe even pre-type 2 diabetes and type 2 diabetes. But they were only able to do that because these samples were time stamped. So it's a really maybe powerful way to use this principle of rhythms in microbes to define health versus disease. It's certainly not solving it. It may not be contributing, but we can definitely use it as a diagnostic. So kind of a cool way to think about it. So I think key takeaways here is that time of feeding coupled with diet composition are the main drivers of microbial community membership. So who's there, and their broader oscillatory diurnal patterns. And we think that there's some innate immune components like antimicrobials that are made by the host that oscillate, and this is occurring independent from our own body clocks. So it's really driven by the types of microbes that we have. And this diurnal expression really depends on the presence of select community members that are selected by the diet. But it ultimately is a very bidirectional communication between our bodies and the microbes that is kind of the secondary driver to maintain these oscillations within specific regions of the gut. So I think, when I think about how do we move this forward? Well, I think the gut microbiome is a key feature of maintaining our circadian network. We certainly can't ignore it. Maybe we could think about using time delivery of prebiotics or probiotics or something that I like to call postbiotics, like these small molecules that are made that can impact our health, right? And promote health and promote the oscillations of specific microbes or host features. And I think, deeply phenotyping ourselves and understanding our sleep-wake patterns, our feeding patterns, our work schedules is really essential to really drill down and, understand how microbes might be fitting into this equation. And as I mentioned on the last slide, diurnal patterns of these microbes are already being used as a diagnostic. So, I think we need to kind of keep that going. And I really don't think it's there's not going to be a one size fits all approach, right? I mean, we know for those of you sitting in the audience, we've got some owls, we've got some larks, everybody's chronotype is different. So we need to kind of think about that and track that and understand it. And I hope healthcare providers will start to think about this too going forward, because I think oftentimes we get asked, well, how do you use the microbiome? Well, I'm not sure we know exactly how yet, but we need to have more information so that we can ultimately manipulate this system to promote health. So with that, I would just like to acknowledge all of the wonderful people who have helped me over the years with these studies. Circadian work takes a team. It cannot be an N of one. Thankfully, we had some self-prescribed owls and larks on our team to keep the machine running over the 24-hour period. My mentor at UChicago was just fantastic and provided me with excellent training. And then my funding sources, which have really, helped me to push the science forward. So, and this is just a beautiful picture actually in Chicago from our building that overlooks Lake Michigan. So when you do circadian work, you get to see some beautiful sunsets and sunrises. So with that, I'm happy to take any questions, and thank you so much.
Andy online, you can read the chat. It's asking about the mini gut, and you can describe that in more detail. That would be interesting. Yeah. So mini guts are really interesting. So what we're able to do, even from human biopsies, so like when you go to get a colonoscopy, or if they're lucky enough to get to the ileocecal junction, they can take a biopsy, and in that biopsy are these crypts that maintain and house the stem cells that are used to make new epithelial cells. So the gut is so interesting because it's probably one of the most dynamic systems in our body and turns over most frequently. So our epithelial cells are constantly sloughing off, even as we sit here.
So, we're able to kind of harness that in that there's a number of stem cells that are maintained in the crypts that then we can apply differentiation molecules to these to kind of drive them down a specific lineage and into intestinal epithelial cells. It's just a fantastic, really interesting way to study the intestine. Now, I will say those mini guts are actually inverted. So, it's hard to kind of treat them because like the lumen is actually inside of that little bubble, right? And we want to kind of get to that luminal space to add our treatments because that's how our guts would see them. So there's actually now new ways to flip them inside out. We can also flatten them out and use them as monolayers. There's people and hospitals that are making banks so that from human donors, so that we can test like IBD, inflammatory bowel disease patients versus healthy, to look at the, like what is contributing to disease development. So they're really, they've become a very, very important tool. Now we can make them from other tissues too. So we can make like what we call hepanoids from the liver. And I think there's a lot of work being done in the brain too.
Are these primary cultures or can you sustain over weeks and months?
They're primary cultures, but they can be sustained over weeks and months. They can also be frozen down and brought back up. So yeah, it's pretty cool.
Other question?
Yeah, I'm sure that like a lot of like commercial poultry growers will alter the light cycles and the time of feeding,
Yep.
and they do it on a huge scale. So can you get any data out of that?
That is such a great point. And when I interviewed here, that's actually one thing I talked about, because you're absolutely right. When birds are heat stressed, especially, they will just turn off the lights because, eating creates more heat. So they'll just turn off the lights on those birds. And I wonder often, like, how does that affect their circadian rhythms? And so, yeah, that's actually something we're trying to work out to do here at UW. We're so lucky because we have a poultry lab in the middle of campus, so we can actually do that work. So it's a really good point. There is some work to show actually that chicks, so baby chicks that hatch, the types of microbes that they acquire can impact how the circadian system develops. So that's another way to think about it too, right? Yeah, it's a really great question.
Not coming from the science bit, but I would assume everybody here is strongly diurnal. So working backwards from the fact that we have circadian rhythms, couldn't just be that influences our weight or.
Oh yeah, I agree. It's a multifactorial situation.
That doesn't seem to be one of the factors.
Well,
so humans are pretty much.
I would agree with you, but I also would disagree a little bit because, for example, people who work third shift, so shift workers. So especially for people who shift back and forth between third shift to first shift and whatever, they actually have higher predisposition for diabetes and obesity. People who experience jet lag, for example, they are also more predisposed to metabolic diseases than people who don't experience jet lag. So I agree. I think that our bodies are meant to be diurnal. We have shifted our environments in certain cases to disrupt that pattern, right? So, and then there's other populations that are really interesting too that we could maybe think about studying, like indigenous populations in Alaska who are thrust into constant darkness or constant light. Animals can sustain rhythms for certain amounts of time without any light dark cues, right? So it's what we call a constant darkness. They will maintain rhythms for a certain amount of time, and then they'll start to kind of free run and do their own thing over time if they're maintained in constant darkness. Now, constant light, actually, animals will die if they are kept in constant light conditions over a long period of time. So yeah, it's.
So 6 months is not a long period of time.
In the span of an animal's life, I would say it can be a long time. For humans, not so much. But yeah. So we always say, well, we don't always say, but people who do have to experience shift work, having a sustained shift. So if you're always third shift, you're less inclined to have metabolic diseases or cancer or other types of things. So it's people who have to shift back and forth that have the most disruption. So yeah, it's a good point.
Yeah, if you use like antibiotics to kind of wipe out somebody's microbiome and then try to introduce some new stuff, will that take, or will they just bounce back to what they used to?
That's a really good question. There's a lot of work going on to kind of understand that, because there are certain individuals who, when they're exposed to antibiotics, they will bounce back to their original state. But there's some individuals that don't ever return back to normal. But what does that mean, right? Like, we don't have long-term data to really support that in people. But it's a really good question. And there is some work to show that probiotic intake after antibiotics or during it is not helpful. Like, it doesn't do anything. So people often ask, like, probiotics, should I take them? Should I not? I'm like, well, probably not going to hurt you, but probably not doing anything either. And I will say one of those reasons is because probiotic off the shelf, we don't really know what's in it, right? We don't know if it's live. We don't know how many bugs are actually there doing what they claim to do. So, I don't discourage people from doing it, but I also say, like, if it makes you feel good, go ahead. But I can't say that it's, doing what the bottle claims it's doing. So I would say prebiotic intake, like taking in high fiber or finding a fiber that works for you, is probably more beneficial than taking in that probiotic, right? So the food sources themselves are the important piece to this. Yeah, I'll take that and then we'll go.
What happens when you get C. diff?
Oh, C. diff is a whole special situation. So for C. diff, Clostridies, they just changed the name of it, difficile. So this is essentially when you have this one specific microbe in your gut that just sort of takes over, and it's like a weed. It just like expands and it pushes everybody else out. And a lot of times you think, well, I'll just treat it with antibiotics, right? But it can be very antibiotic resistant, and it just will never go away. And one of the common treatments for this is fecal microbiota transplantation, right? So taking microbes from a healthy person and putting them into a person with C. diff. And sometimes a single treatment can actually recover that microbial community membership. Not for everybody, but for some people. And so kind of the thinking is that we need to add things that can compete out the C. diff and break that loop.
So C. diff is just one bacteria that multiplied like crazy.
It's like one species of bacteria. There might be different strains at the strain level within that, but yeah, it's one specific bug that does that. Yeah, and then we're trying to understand why. And it actually, so our bodies produce what are called bile acids. And bile acids classically have been thought to be there to help with absorption of fats, right, from the diet. But it seems that C. diff has a really unique way of interacting with our bile acids, which might actually sustain it in that community too. So we're trying to understand that right now too. Not in my lab, but in other labs on campus. So yeah, C. diff is a really special, interesting, dangerous case. Yeah, good question.
I was curious about research that has been done on children that grow up on farms versus kids that are grow up in the city. And I know that asthma and other kinds of diseases differ between those two groups, but are there other things that have been studied?
Yeah, there's a group actually at University of Chicago. Her name is Carol Ober, and then another investigator, Ann Sperling. And then there's some work actually being done here at UW looking at Amish populations, even non-Amish, but farm populations relative to non-farm populations, specifically in children, and looking at asthma outcomes. And there are very unique microbial properties for example, in Amish children on farm who typically don't develop any asthma, right? Even relative to those who are on farm children, there's unique populations present in the Amish community specifically. So that says a lot about, you know, how these children are raised, what they're exposed to. So yeah, even type of farm can impact the types of microbes that are present and how that associates with disease like asthma, for example. And so we're interested in that from a metabolic disease perspective too, because you don't see obesity in some of these on-farm populations, whereas city kids, for example, have higher incidence of metabolic diseases, even at younger ages. So how do microbes sort of set up our metabolic set point? It's a really great question. I don't have a precise answer, unfortunately.
The next week's speaker is going to be Dale Scholler. He's collaborators at the University of Chicago also.
Yeah.
Are you familiar with Dale's work?
Yeah, using isotopes. Yeah, so actually I know the investigator he was working with, Esra Tassali, and they were trying to use stable isotope measurements to look at metabolic outcomes dependent upon sleep and sleep disruption. Yeah, really interesting stuff.
It sounds like his cousin worked with this your group of circadian sleep.
Yeah. I don't know if Dale and Esra have looked at microbiome specifically, but yeah, their stuff is really, really interesting. I kind of want to sneak in the back door to listen to what he has to say.
It'll be zoomed on that same.
Okay. Yeah, I would like to hear that.
I have a question related to colonoscopies, when they make you drink that gallon of stuff to wipe you out.
Yeah. The go lightly prep.
And do some people not come back from that?
That's a great question. There's a lot of ongoing research trying to understand that. And colonoscopy preps are like, I mean, they're terrible for those who have to do them.
We all know.
But yeah.
You don't need to tell us.
But they're so important for the gastroenterologist that's doing your colonoscopy, because I will tell you, if you're not prepped appropriately, they won't do the colonoscopy. It's a failed colonoscopy. So that's a really good question about whether or not the microbiota recovers and how it recovers. I will say that a lot of stuff that we have done where we've been able to go into actually biopsy, we actually have unprepped people. I served as a control for unprepped colonoscopy once because we were worried about that, is what happens if you prep people to the microbes. You have less biomass that's present, right? And then also you could be changing the microbial community membership just by flushing everything out. Yeah. But I personally have not delved into the studies for recovery following colonoscopy of the microbiota specifically, but.
Is there the same biomass in the small intestine as the colon?
No, actually we have far fewer microbes from mouth to anus. So microbes in the mouth are very small numbers, in the duodenum, which is right after the stomach, very small numbers, and it just incrementally increases the further down the gut you go. And then ultimately the colon has the highest biomass and highest numbers, as well as the most rich community membership as well. So yeah.
Other question?
See you next week. Thank you very much.